So in this session, we'll cover the science of human genome editing. So this is a picture I downloaded from the website. Was the first summit. And today we have 4 wonderful speakers. They're going to tell us about the story of mostly CRISPR and its potential application. Most critically, perhaps, things happen between 2015 and then today. So we have a 3 years gap between the first summit and the second summit. And most of the advances happening during that 3 years will be covered by 4 wonderful speakers. So the first one is Feng Zhang from the Broad. Second one is David Liu from Broad 2. And Angelo Lambondo from San Rafael, and then lastly, Jennifer Doudna from Berkeley. So without further ado, I'd like to invite Fang to come to the podium.
All right. Good afternoon. It's really a pleasure to be here. I was at the first Genome Editing Summit 3 years ago, and I thought that was a really great meeting, and there was a lot of really insightful discussions happening, and certainly very glad and honored to be back here. I was asked to give a primer on gene editing, so I'll tell you a little bit about how gene editing works and what are the different modes of application that are being contemplated and developed. So one of the motivations for gene editing has come from the rapid accumulation of genetic data. The first human genome was sequenced more than 10 years ago, and the cost for sequencing human genome has dropped precipitously over the past decade. And so now we can sequence the complete genome of an individual for under $1,000. And because of that, we can now start to collect many individuals' genomic information. In fact, there's large studies underway now that are trying to get genome sequencing data for 100,000 or even up to 1 million individuals. So by comparing and analyzing the differences across large populations of patients and healthy individuals, scientists are beginning to identify many different genetic signatures that may play a causal role in disease. And so some examples of genetic causative mutations include the ones that cause sickle cell disease or cystic fibrosis and also eye degeneration. So these are causative genetic mutations. And because of that, one of the thought is, if we know what caused the disease, why don't we just go back into the genome of these cells and remove them and put in the sequence that doesn't cause disease. So that's where genome editing comes in. Genome editing has been under development by a whole community of scientists over several decades and has undergone several iterations of technologies, sort of becoming easier and easier to reprogram, to retarget new DNA sequences so that we can change DNA sequences in the genome practically at will. So how does genome editing work, and what are the differences between genome editing and also some of the other technologies that provide intervention at the DNA or the RNA level inside the cells? So before we dive into the mechanism of genome editing, I thought I'll tell you a bit about the genome first. So the genome consists of different chromosomes, and each chromosome is a long string of DNA sequence. DNA has a very simple alphabet. It's just got 4 letters, A's, T's, G's, and C's. And if you zoom in to a section on the chromosome, you will simply see a long string of letters. Looks like this, CC, TGC, and so forth. Just by looking at the sequence, it's hard to make sense of what these sequences mean. And so scientists who have worked on trying to map the functional elements of the genome have begun to shed light onto the structure of how the genome encodes information. And so one analogy of how the genome encodes information is we can think of it as a long document. So just like a written document, the document contains sentences, and these sentences have structures, they have elements, different words that come together form a sentence. And so the genome, in its own DNA document form, consists of different sentence structures that are more or less gene cassette units. And so that long string of DNA sequence can then be annotated and classified based on the functional elements. And each one of these functional elements is shown here as one of these cassettes. So if we take this analogy a little bit further, each one of these gene cassettes can be broken down into several different elements. So there are things called promoters, enhancers, genes, and also terminators. And to compare with what a spoken sentence might be or written sentence might be, a promoter is akin to a verb. It directs the action of the gene. And the gene is the noun. That's the main principal character in that cassette. And then there are enhancers, which are adjectives. It tells the gene to express a lot, or to not express very much, or to not express at all, or have other alterations to the way that it's being produced by the cell. And then there are the punctuation marks, which are the terminators. It tells the cell when to stop producing that gene product. So just like a written document, a mutation in the genome is like a typo in that written document. And so there can be many different forms of typos. And so consider this very simple sentence here, twinkle, twinkle, a big star. So of course, here there's a typo. The sentence normally sings twinkle, twinkle, little star. And one way to be able to fix the sentence is to be able to put the correct word back in. And so one of the earlier methodologies of perturbing or correcting molecular information in the cell is through gene therapy. And so gene therapy, what it does is that it puts the correct gene into the cell, but it doesn't remove the incorrect gene. And so what you end up with is a sentence that looks like that. So this is gene therapy on this sentence. You get twinkle, twinkle, little big star. Now, clearly, gene therapy alone is not able to fully restore the meaning of the sentence. And so then another methodology called RNAi, or gene knockdown, can be used to be able to get rid of things. And so if you perform RNAi on this sentence, this is the end product. You get twinkle, twinkle, star. Now that a word is missing. And so it's because of this, gene therapy adds a word, RNAi knockdown removes a word, but neither put in the correct word or replace with the correct word, then this is where gene editing comes in. Gene editing provides the ability to fully restore the meaning of a sentence within the context of the genomic document. So how do we then use gene editing to actually achieve changes at a genomic level? If this was really a document, you can open up in Microsoft Word and open a search box, find where the typo is, backspace to delete, and then to type in the correct word. So the analogous search and replace function in the context of the genome was then the latest iteration came from a bacterial immune system called CRISPR. CRISPR, in fact, has already been used to produce different strains of bacteria to become immunized against viruses that are used to produce yogurt and other dairy products. And it is this same immune system that we can transfer into human cells to then be able to target a specific mutation in the genome and then facilitate gene editing within the context of the genome. So here's a video that kind of summarizes how gene editing works. The blue glob is a protein called Cas9. It's from the CRISPR system. And you can pair this protein with a short RNA that's been designed to have homology to a sequence in the genome that you're trying to edit. This complex, once it's introduced into human cell, can search along the DNA and try to compare the RNA with the DNA. When comparison is established or similarity is established, then the enzyme will get activated and it will cleave the DNA to form a double-stranded break. So this is the cursor in the genome. Wherever you place it, that's where editing can happen. Now, there are two ways to repair this, and both depend on endogenous cellular repair machineries. The first one just re-glues the two ends together, but doing so in an error-prone way that can inactivate a gene of interest. Alternatively, you can also provide a synthetic piece of DNA that carries homology to the two broken DNA ends. And the homology direct repair process can then recombine that new piece of DNA into the genome, in that process also incorporating a new piece of DNA sequence that you want in a very precise way. And so these are the two main ways that we can go about achieving gene editing. So the same technology is now underway for development to be able to treat different diseases. Two of the modalities for deploying gene editing include ex vivo-based therapy, where especially cells in the blood system can be removed from the body, modified in petri dishes, characterized to make sure that they have been successfully edited, and also precisely edited, and then reinfused back into the patient to be able to treat diseases like sickle cell or engineering immune cells to be able to treat cancer. And then the second modality targets tissue that cannot be removed and replaced back in the body. So for disease that affect the muscular system or the liver, the brain, these gene editing reagents can be directly introduced into the body, targeting to specific tissues, and then achieve editing of the genome in vivo in the body of the patient. So these are the various ways that gene editing hold promise for developing gene editing through the editing of somatic tissue and correcting disease-causing mutations. And that's the primer on gene editing. Thank you very much.
That was wonderful. Okay. So now I introduce David Liu from the Broad, and he's going to talk about the base editing.
All right, thank you. I'd like to begin by thanking the organizers and describing what an honor it is to engage not just with other scientists, but of course with a much broader community represented by this summit. So thank you for the opportunity to speak here. I was asked to present the science of base editing, its development, and its early application. And so I'll begin with some important disclosures that I'm co-founder and consultant of 3 genome editing companies, Beam, Editas, and Pairwise, as well as SAB member for WuXi UpTech. The inspiration behind base editing is really this pie chart, that most of the human genetic variants that are associated with disease are point mutations, the simple change of one base pair for another base pair. But the irony is that standard genome editing methods, including the use of CRISPR-Cas9 nuclease, have difficulty correcting these single nucleotide polymorphisms, these point mutations, efficiently and cleanly, meaning without an excess of undesired byproducts. And the reason is that if your goal is to correct a point mutation and you approach the problem using the molecular scissors, that is a programmable nuclease like CRISPR-Cas9, you begin by making a double-stranded cut in DNA near the site of the mutation that you wish to correct. The cell's natural response to that double-stranded cut, most of the time, under most circumstances, in most cell types and cell states, is to perform an end-joining process that can result in the insertion or deletion of small numbers of bases, or sometimes large number of bases, as well as translocations and other cellular responses. But if the goal is to correct a point mutation or install a point mutation, these end-joining outcomes are actually undesired byproducts. If the goal instead is to disrupt a gene or to insert DNA, they can be helpful. But for the purposes of correcting a point mutation, they are undesired byproducts. And so instead, we rely on, if you begin by making a double-stranded cut, you have to rely on processes like homology-directed repair, which allow you to replace the DNA sequence around the double-stranded break with DNA sequence that you provide in an exogenous template. The challenge of HDR, as is well known to this community, is that its efficiency tends to be pretty modest, especially in undividing cells, and most of your somatic cells are not actively dividing. And therefore, the take-home message from this slide is that the challenges of correcting a point mutation have actually limited the targets that we can choose to study or perhaps treat using genome editing methods if we limit our technologies to programmable nucleases. So base editing is a different approach that performs chemistry directly on a target base in the genome of a living cell or organism. Base editors build on the seminal discovery and characterization of CRISPR enzymes that many of the speakers here and others in the community have reported. But instead of serving as molecular scissors that cut DNA, base editors have been disabled so that they can no longer make double-stranded cuts in DNA. I liken base editors more to pencils rather than scissors, because while they retain the ability to bind to DNA in a guide RNA programmed manner, instead of cutting the DNA, they have other proteins which allow the base editor to directly catalyze the conversion of one DNA base to another DNA base. In the first form of base editors that we reported in 2016, the entity that actually performs the chemistry on DNA is a cytidine deaminase enzyme. This is an enzyme that catalyzes the conversion of C to U. So a GC base pair gets opened up by this catalytically impaired form of Cas9 within a small single-stranded bubble created by the Cas9 protein guide RNA complex. The C gets converted directly into a U. And then our work isn't done. At this point, we have a GU mispair, and in order to convert that GU mispair into an AT base pair, the desired goal of this class of base editor, we have to carefully manipulate the way that this UG intermediate is resolved by the cell. So the purple protein then steps into action. It's part of our current generation of base editors, and it helps to protect the uracil intermediate from being removed by the cell in DNA. Once that UG intermediate is protected, then a current form of our cytosine base editors called BE3 nicks the G-containing strand of the UG mismatch, which tricks the cell into fixing that strand using the uracil, using the U as a template. Okay, so nicking the G-containing strand stimulates the cell to replace this UG intermediate with a UA and eventually a TA base pair. So what we've done is convert a GC base pair into an AT base pair without inducing a double-stranded break. And the efficiency of this process, as you add in each of these components into the base editor, tends to be substantially higher than what we observed using Cas9 nuclease and an HDR template. And in addition to higher efficiency, the product selectivity, how cleanly one can edit cells, tends to also be higher when we perform base editing than if we perform cutting followed by HDR. So shown here are human cell sequences in cultured human cells that we've treated with Cas9 nuclease and an HDR template, along with a guide RNA that programs and a template that programs the insertion of the replacement of these bases with the green bases. And you can see that you get some of the desired edits, some of the desired point mutations, but you mostly get these deletions and occasionally an insertion. If you take the same cells and replace Cas9 and the HDR template with a base editor with the same guide RNAs as before, instead you get this kind of product distribution in which most of the products that are edited are the desired product. So these features of base editors have, coupled with the vibrance of this community, have caused them to be used rapidly by the genome editing community. So even though our first base editing paper was reported only about 2 years ago, there have now been almost 100 papers published using these tools to perform point mutations for studying and even treating cell and animal models of genetic disease. A second class of base editor that we reported about 1 year ago now is a different type of base editor that converts AT base pairs to GC base pairs. This particular new class of base editor, called the adenine base editor, is of particular relevance to living systems because it turns out that the distribution of point mutations in living systems is not evenly distributed among the 6 different ways you can change one base pair to another base pair. Instead, almost half of point mutations in living systems, including pathogenic human point mutations, are of the sort in which a GC base pair has become an AT base pair in the mutated form. So to correct those mutations requires developing a new class of base editor that converts AT base pairs to GC base pairs. One strategy to do so, a chemical strategy, is to recognizes that if you deaminate the exocyclic amine of adenosine, you get inosine, which is a base that pairs much like G in DNA and RNA polymerases. So in principle, one could take the same strategy that we use to develop the cytosine base editors and apply them to adenosine. We would open up DNA using a catalytically impaired form of Cas9 at a target site, and within the single-stranded bubble of DNA created by Cas9, the target A would get converted into an I. We would nick the T-containing strand to trick the cell into replacing that T with a C, and now you've converted an AT base pair into a GC base pair. The problem with this idea, the main challenge, is that there is no naturally occurring enzyme that deaminates adenine in DNA. So unlike the case with the cytosine base editors, in which we could use enzymes that nature provided to convert C to U in DNA, there is no known such enzyme that does so in DNA. So we actually evolved a DNA adenine deaminase enzyme, beginning with one that performs a similar reaction on RNA. I won't go into the details in the interest of time, but suffice it to say that we developed a kind of laboratory selection in which mutants of this tRNA deaminase enzyme that accept DNA have a survival advantage. They enable antibiotic survival. Then we could take the mutants that survived and transplant them into a base editor architecture designed to work in mammalian cells. And then, to make a long story short, I'll just jump to the answer. If you end up performing this laboratory evolution, you end up with mutations shown here, which collectively allow this enzyme that normally deaminates A in tRNA to now work on A in DNA. In addition, we broadened its sequence context requirements so that it no longer has to require the exact sequence of the surrounding bases that were present in the natural tRNA substrate, but instead accept a wide variety of bases in DNA. And so while we started with 0% editing, as you proceeded through these rounds of evolution, by the end of 7 rounds of evolution and engineering, we had developed a base editor that efficiently converts AT base pairs to GC base pairs in a wide variety of sequence contexts. And like our original class of base editors, the adenine base editor has also become rapidly adopted. It's only, again, been about a year since we published the first paper, but a number of papers have already been published using these base editors in a variety of organisms. And AdGene, the plasmid nonprofit distribution service, has distributed more than 6,000 base editor plasmids to date to researchers around the world. So efforts by our lab and by many other labs in the community to expand the scope of base editing, I've summarized on this slide. We've worked to improve the purity of the products that come out of base editing and to enhance base editing efficiency. Beautiful work by Lucas Dow's lab has also arrived at solutions to this challenge. We and others have maximized the genome editing breadth of base editing, taking advantage of naturally occurring or laboratory engineered or laboratory evolved variants of CRISPR enzyme that allow base editors to target a larger and larger fraction of the genome. We and others have also worked on reducing undesired base editing events, including editing of nearby Cs or As that are not the target base, and of course reducing off-target editing at sequences that are similar to but not identical to the desired target sequence elsewhere in the genome. Keith Jun's lab and Fun Zhan's lab have both contributed to this problem, as well as Jennifer Doudna's lab. And then finally, we have begun to study, and other researchers have also reported positive results establishing that base editing can actually occur efficiently in post-mitotic, non-dividing somatic cells in vivo, in contrast with some of the challenges that face the use of Cas9 nuclease and HDR. So I'll end with just a couple slides on some new data. This is early data, unpublished still, but that integrates many of these concepts and many of the other advances that the field has worked hard on to show how we're trying to use base editing, and I think many other labs are using base editing to address animal models of human genetic diseases. So progeria is a rather well-known genetic disease. A common form of progeria is caused by a single CG to TA point mutation in a gene called lamin A. The consequence of that point mutation is not to change the coding sequence of the gene. It's actually a silent mutation, but is instead to create an early RNA splicing site that causes the early splicing of exon 11 so that you end up losing 50 amino acids of coding sequence from the resulting mRNA. The protein that is translated from that mRNA is called progerin, and it behaves in a dominant negative way to poison nuclear membrane structure, and this results in premature aging. Cardiovascular problems are typically a cause of death, and the kids who have progeria, while mentally fairly normal developmentally, typically have lifespans of only 13 or 14 years. Because progerin is actually poisoning their cells, this is a case where gene therapy approaches of just making more of the healthy gene and perhaps even using nucleases to cut and disrupt the pathogenic allele face interesting and I think difficult challenges. So we were particularly interested in using base editors to try to directly correct the cause of this disease in patient-derived cells and eventually in animal models of this disease. So we took fibroblast cells derived from two progeria kids, two progeria patients, and infected those fibroblasts with lentivirus that expresses a base editor programmed to directly reverse the mutation from this TA base pair back into a CG base pair. We were encouraged that in these cells in a dish, we were able to achieve 40 to 80% editing, depending on the amount of time and the sample, directly correcting this causative mutation back into the wild-type sequence with a very low percentage of detectable insertions or deletions, which again, in this case, would be potentially undesired. And then finally, we have begun to do pilot studies on taking these base editor technologies and applying them to the mouse model of human progeria. This is a mouse model that Francis Collins, currently the director of the NIH, developed and reported in 2006. We used AAV as a delivery vehicle, which is, of course, especially therapeutically relevant because AAV is used in the clinic already. And we developed a split A-base editor optimized with a certain variant that allows it to target the right position of this gene, put it into two viruses because it's too large to fit into one virus, and injected a mouse at 4 weeks with a single injection of this pair of viruses. And at the end of 8 weeks, we assayed it at the DNA, RNA, and protein levels. And this is just a pilot experiment where, to be honest, we weren't expecting very positive results, but we were pleasantly surprised that in this mouse, following a single treatment, we observed up to about 60% editing in some organs of the actual pathogenic allele that causes progeria. That editing was extensive enough that when we assayed the RNA for correct splicing versus missplicing, we saw that we could go from predominantly misspliced RNA to mostly correctly spliced RNA in the tissue derived from these animals. And then finally, at the protein level, gratifyingly, these results also translated, where we can see that the untreated mice make mostly the middle band, which is the toxic progerin protein, whereas our treated mice make mostly the correctly spliced protein. Okay, so to summarize, base editing is a newer form of genome editing that enables us to directly convert one base pair to another base pair without making double-stranded breaks or relying on homology-directed repair. We now have developed 2 classes of base editors that collectively enable the installation of all 4 types of transition mutations. Of course, there are still 8 transversion mutations that we and others are working on. You can make those mutations with high ratios of desired editing outcomes to undesired indels in many different cells and organisms. And the field has extensively developed engineered and evolved forms of base editors, new Cas9 PAM variants, and new in vivo delivery technologies that have really begun to expand the capabilities of base editing and increase their in vivo applicability. Finally, that leaves me to thank a very talented and hardworking group of students. I'm grateful to my collaborators on the Progeria project, Jonathan Brown and Charles Lin. Grateful to these agencies for funding, and I'm happy to answer any questions. At the end of the session.
Thank you, David. So next speaker is Angelo Labondo from San Rafael.
Thank you. So good afternoon to everybody. It's a real pleasure to be here and an honor. I've been asked to briefly describe the principle of gene therapy and give you an examples on how you can use this powerful technology in gene therapy. Very briefly, I've been introduced also very nicely by Feng, some basic concept of gene therapy. I'm referring to, in particular, to gene transfer in somatic cells, and most of the time stem cell, somatic stem cells. In this case, we often use viral vectors, thanks to the ability of these vectors to deliver their genetic information into a cell. And this genetic information is a functional copy of the missing gene in this particular cell. And these vectors, most of the time, if we are dealing with retroviral or antiviral vectors, have the ability to semi-randomly distribute their genetic information into the chromosome. And this is a plus for the technology, as it allows to achieve long-term stable correction of the disease. And indeed, this approach has been used since many years in several clinical trials, providing evidences of effective gene transfer and gene correction in several disease conditions. There are, however, some limitations that relates to the use of these vectors. First of all, as I said, they semi-randomly distribute the genetic information into the chromosome. They may cause some risk of genotoxicity, meaning that the vectors and the regulatory elements present in the vectors may interact with the regulatory elements of endogenous gene, nearby endogenous gene, deregulating their expression. Sometimes it is difficult, actually, to recapitulate faithfully the expression, endogenous expression of the corrective gene in the vector because of constraints in their dimension. And lastly, not least actually, it might well be that the vector can be immunogenic. I don't have to say to this audience that actually in the last 20 years, new technology have emerged that can allow to actually not really to randomly correct the genes, but to precisely modify the genome. And we are dealing with artificial nucleases. Well, very briefly, what happens is that you can use either zinc finger nucleases, tau nuclease, or CRISPR-Cas9 system to induce a DNA double-strand break into the genome by programming this protein to recognize a specific sequence. The break, as said before, can be repaired by several pathways. One of these pathways is non-homologous unjoining. And this pathway that seals the break often cause insertion or deletions that can cause stop premature codons and thus gene knockout. The second pathway instead is homology-directed repair, is a faithful pathway mostly active in the SG2 phases of the cell cycle. And this pathway can be actually harnessed by the investigator by providing to the cell what is known as exogenous template, a sequence of DNA that contain the desired sequence plus flanking homology to the site that have been recognized by the nucleases. And the homology-directed repair basically copy faithfully what is embedded in this homology inside the nucleases target site. And this can be used to correct single-point mutation in the genome, but can be used also to insert long stretches of DNA. I'd like to give you an example on this. We are dealing here with a genetic defect that is caused by mutation in this interleukin-2 receptor gamma chain gene, which is placed in the X chromosome. Mutation in this gene are causing a severe combining immunodeficiency. These patients lack basically T cell, NK, and have impaired B cell activity. So basically, most of the immune harms of these patients are affected by this disease. What you can do in this case is to design or program a nuclease targeting an exon of the gene, in this case exon 5, which is a hotspot of mutation, and induce a break in the exon 5, and at the same time provide to the cell this exogenous template DNA that I mentioned before that contains homology to the site that have been recognized by the nucleases, but also a partially corrected cDNA, which is basically a piece of DNA which contains all the exons which are located at the site of the nucleases, downstream of the site of the nucleases. So basically, this partially corrected DNA in this specific case contain exon 5, 6, 7, and 8 of the L2 receptor common gamma chain gene. In this specific case, we co-targeted together with the corrected cDNA also a reporter gene, GFP expressing gene. And the outcome of this process is insertion of the corrective cDNA and GFP within the L2 receptor common gamma chain gene. Here comes the beauty of the approach, because you can use this corrective cDNA basically to correct a broad spectrum of mutations, so basically all mutations which are located in this gene from exon 5 until exon 8. At the same time, you are physiologically expressing the gene that have been corrected thanks to the endogenous regulatory elements of the L2 receptor common gamma chain gene. On the bottom, you see an example. These are T primary lymphocytes from a healthy male donor. Again, the gene is located in an X chromosome, and you perform the gene targeting procedure. You can see on the flow cytometry plot on the right. And what happens is that a fraction of the cells is unaltered, so they remain with their own L2 receptor common gamma chain gene, while another fraction, 30% of the cell, in one single treatment actually express now the corrected cDNA, but also the GFP. So this has been done in T lymphocytes. Actually, the main target for this disease, we are dealing with ex vivo gene therapy, are non-T lymphocytes, but actually hematopoietic stem and progenitor cells from these patients. And we struggled a lot, actually, to develop protocols that can efficiently, rather efficiently achieve editing in these cells. As mentioned before, some cells are more refractory to homology-directed repair than others. In this specific case, we were able, by, again, dealing with the technology, the delivery technology, and the timing of delivery, to achieve fairly good efficiency of gene editing in this relevant cell population, roughly 40%. And we also tested the ability of the edited cells actually to repopulate a host, in this case a mouse, in order to understand if the procedure was harmful to the cells. We were happy actually to notice that basically when you transplant these cells into a mouse, they are able to home into the bone marrow of the mice and produce the hematopoietic human lineages in this animal. To conclude this part of targeted genome editing, I'd like to say that the beauty of this system is that you're taking basically two birds with one stone, because you are reconstituting the gene, but at the same time you are achieving physiological expression control of the corrected gene. It's a plus in gene therapy. You overcome also the risk which is associated with a random distribution with vectors, with lentiviral vectors or gamma retroviral vectors of the information in the genome of the cell. There are still challenges to the technology. As mentioned several times, efficiency is not always high enough to achieve phenotypic gene correction at the end. So a lot of efforts are now ongoing in terms of improving the efficiency of the technology, not only in non-proliferating but also in proliferating cells. This concept also is linked to the yield of the edited cells. Although we may achieve fairly good efficiency, 20–30% of gene editing, we need to purify these cells in order to achieve phenotypic correction. And there is also the need to characterize the specificity and immunogenicity of this cell. Still on gene editing, actually, I'd like to share with you some idea that we got, and pertains the fact that not only you can correct single point mutations into the genome or insert this corrected cDNA, but you can exploit gene editing to confer novel biological properties to a cell. And in particular, we are aiming here to generate a universally transplantable cell because it's clear that there is a paucity of tissues or organ when dealing with organ transplantation. And one of the biggest problems is actually to overcome an immunological barrier that is due to the fact that most of the time donor cells express highly polymorphic molecules on the cell surface. They are named HLA A, B, or C. And these highly polymorphic molecules in the donor cells on the tissue can be effectively recognized by allogeneic immune cells. Recipient, in this case, recipient TCL, through interaction of this molecule, HLA class 1 ABC and the TCR. It will happen that the immune system will lyse the donor cell. A way to hide the donor cells of the tissue to allogenic T cells is basically to inactivate the HLA, highly polymorphic molecules, for instance, by using gene editing and disrupting the beta-2M gene, which is a constituent of this polymorphic cell molecule. By doing so, it is an example, we actually disrupted the beta-2M gene in a pluripotent stem cell with the aim of obtaining universal donor pluripotent stem cells that can be differentiated in tissue of relevance. If you disrupt beta-2M, not only you lose the HLA class 1 highly polymorphic molecules, but these cells became basically invisible to the allogeneic T cells. You can see in the graph on your right. Unfortunately, the immune system is smarter than us and developed another arm of immunity, that is the NK cells. So these cells are programmed in our body to recognize cells that are missing the polymorphic HLA molecules. And indeed, on your right, you can see again that if you put the beta-2M disrupted cells together with the NK cells, the NK lyse effectively these cells. So we and others come with this idea, so basically to disrupt beta-2M by inserting into beta-2M HLA molecules which are invariant and are often present in the maternal to fetal interface in order to avoid immune rejection in the specific system. And here again comes the beauty of gene editing. You disrupt a gene, but at the same time you exploit the gene that you have disrupted to express totally new molecules that should impart an advantage to the cell. On the right, you can see the efficiency by which we can achieve this gene editing disruption, integration, roughly 8%. And if we sort the HLA-E expressing cell, the beta-2M negative cells and the wild-type cells, and expose them to CD8, allogeneic CD8 or NK cells, we can notice that in case of CD8, both the HLA-E and the beta-2M disrupted cells resist to T-cell attack, while in the case of NK cells, the beta-2M negative cells can be easily disrupted by the NK activity, and the beta-2M negative HLA-E now positive cells are resistant to NK activity. With that, I'd like to conclude the part on gene editing and briefly switch on another field of research that we and others are developing and deals with gene silencing. As you know, as I said before many times, so the scope of gene therapy goes well beyond gene replacement. There are many diseases that can be treated by silencing a gene. For instance, disease caused by dominant negative mutations, repeat expansion disorders. People have also postulated and used actually many technologies, gene silencing technology to treat infectious diseases. And there are also more classic inherited monogenic diseases like hemoglobinopathies that can be treated not by replacement, by gene silencing. Several technologies are around. You're probably familiar with RNA interference. In these specific cases, you are decreasing the expression of the messenger RNA of the gene. In order to achieve stable RNA interference, you need to stably express this molecule or deliver them continuously. We mentioned several times gene inactivation. You can disrupt the gene. We mentioned also the fact that this disruption may cause a severe DNA damage response into a cell that can apoptose, or in the case of primary cells, cause differentiation of the stem cell. We and others exploited targeted epigenetic silencing to achieve gene repression. The epigenome is something that is not yet described in this meeting. It's basically a code that is present on the chromatin, and it takes activation or repression of gene expression. Here there is, in my opinion, a beautiful example of this, a mechanism by which in the early phases of our development we silence endogenous retro elements that are present in our genome, what people have also named before junk DNA in our genome that is not actually junk DNA in this specific case. This process of repression is activated by classes of molecules that can, like the artificial nucleases that we are using, can specifically recognize sequences into the genome, and in particular the endogenous retroviral sequences. And these molecules activate a plethora of other proteins that modify the code of the histones onto which DNA is wrapped around towards, in this specific case, a repressive state. Not only these molecules modify the histone code, there are another family of molecules, the DNA methyltransferases, that modify the primary DNA sequence, locking the initial repressor imposed by the Krabs-Finger protein, the original family of proteins that recognize endogenous retroelements on the genome. So the players of targeted epigenetic silencing are, again, engineered proteins, engineered transcriptional repressors. They do contain a custom-made DNA binding domain, a catalytically inactive Cas9, for instance, Zinfinger protein or tail, and these DNA binding domain are fused to effector domain, most of the time coming from naturally occurring transcriptional repressors. And here on the left, there is the list of some of these naturally occurring transcriptional repressors that we and others picked to generate engineered transcriptional repressors. To give you a very rapid example on the functioning of this technology, in these specific cases, we are targeting, again, the beta-2M gene, and in particular the promoter region of the beta-2M gene. Here we are not inducing DNA disruption, we are modifying the code, the histone code of the gene towards a repressive state. And by a simple transient expression of these engineered transcriptional repressors, we can achieve fairly good efficiency of silencing, which is around 80%. The beauty, as you can see on the graph on the left, is that the expression of the ATRs is transient, but then the cells inherited the epigenetic modification imposed by these repressors, stably maintaining repression of the gene even after cell proliferation. This is a possible therapeutic application of this epigenetic silencing. We are dealing with a gene that is named PCSK9 that has been positively and negatively involved with familiar hypercholesterolemia. If you have gain-of-function mutation of this gene, you may have a familial form of hypercholesterolemia, and this hypercholesterolemia may cause, at the end, coronary heart disease. As you can see here, again on the left, by a single treatment, we can achieve roughly 90% of permanent epigenetic silencing of this gene. The mechanism of action is in line with the original mode of action, actually, of these factors during development is basically DNA methylation, and at least for the beta-2M gene, the platform is very specific. I'd like to conclude this by providing a brief summary of this technology. we basically repurpose this beautiful silencing machinery to obtain a hit-and-run technology by which these engineered transcriptional repressors are transiently expressed into a cell, and they stably modify the epigenetic code of these cells, and the cells inherited these repressive states throughout cell division. This technology apparently is resistant to external stimuli, so you can impinge, try to reactivate the gene by providing stimuli to the cells, but the epigenetic modifications are very stable. And as I mentioned before, in order to inactivate the gene, you don't need to activate the DNA damage response. I'd like to conclude by acknowledging all the people that are involved in this work, especially the PhD and postdoc in my laboratories that are working on the epigenetic editing technology, and many collaborators in the San Raffaele Institute and abroad for the different projects we have in the lab, and the funding agency, and you for your attention.
Thank you, Angelo. So our last speaker is Professor Jennifer Doudna from Berkeley. Please.
Well, good afternoon, everyone. It's a pleasure for me to join my colleagues here in this session on the science of human genome editing. You've heard from Feng, David, and Angelo about some of the incredibly exciting applications of RNA-guided enzymes. And what I thought I would do in my 15 minutes is tell you a little bit about where the field is going, and in particular, how fundamental research continues to drive the underlying technology and our understanding of how we can deploy these enzymes in applications that include therapeutics. So first I wanted to point out that I'm part of a number of companies now that are using genome editing for different kinds of applications, and those are listed here. They're also on my website if anybody's ever wanting to know this in detail. And then I just wanted to point out that the field of genome editing, going all the way back to the earlier technologies that Dana Carroll talked about this morning, have always relied on an underlying appreciation of how enzymes interact with DNA. And in the case of the CRISPR-Cas9 system, this is a technology that, as you heard earlier, came from studying how bacteria fight viral infection using RNA-guided systems that allow detection and destruction of viral DNA. In our case, we were investigating a system that involves a single enzyme called Cas9, and in collaboration with Emmanuel Charpentier's lab, we published work showing that this enzyme can be programmed with RNA molecules, allowing it to interact with segments of DNA, 20 base pairs in length, leading to a blunt cut that gets introduced into a DNA molecule, including a genome, if that sequence occurs in a genome, triggering cells to repair the broken DNA, and in the process, introduce changes that allow editing. And we've heard several talks today already about the interesting ways that that kind of editing happens, and ways that this RNA-guided platform can be used to manipulate genomes in ways that don't necessarily involve DNA cutting, as you heard from David Liu and also from Angelo. So what's happened over the last 6 years is that these RNA-guided enzymes, primarily Cas9, have been adopted for a wide range of applications. And this spans not only applications in clinical medicine, but also in research, in healthcare, agriculture, diagnostics, and synthetic biology. And so I just want to point out that there's been a lot of exciting work going on in other fields that frankly also informs on how these kinds of systems will be applicable in biomedical applications. So when we think about how to use an enzyme like Cas9 for therapeutics, which is of course the topic of this conference, I think there's some important things to think about that have come to light over the last couple of years as people have begun moving closer towards clinical trials. for Cas9 in human systems. First of all, the most widely used enzymes for genome editing actually come from pathogenic bacteria. And Matt Porteous and others have shown that this leads to, in many cases, pre-existing antibodies against these proteins, because all of us have been exposed at one time or another to these pathogenic bacteria, but also to the potential for an immune response to proteins that are being introduced as foreign bodies into human systems. So that's one consideration. And secondly, the enzymes that are used right now for genome editing really come from two classes of enzymes. Cas9 is one, and the second one is called Cas12a, or CPF1. These proteins tend to be quite large, and that is a complication for delivery, how we introduce these molecules into cells for therapeutic applications. And finally, there's a desire to avoid genome damage that could occur if there is sustained activity of these proteins over time in a clinical setting. We don't want to have enzymes that are very good DNA cutters having the ability to cut the genome many times and have the likelihood of introducing an undesired change to the DNA. And so for these reasons, our lab, and of course many others, have continued to investigate the natural systems that have evolved in microbes over millennia that have created a large number of diverse enzymes that have RNA-guided properties but have other differences from each other that might make them interesting for therapeutic applications. And I want to just tell you two very short stories today that illustrate how one does this type of research and the kinds of discoveries that lead to new and enhanced technologies, we hope, in the end. So the first little story is about looking for new kinds of CRISPR-Cas enzymes from non-pathogenic bacteria. And we've had, in my lab at Berkeley, we've had the great fortune to be working closely with a wonderful team of scientists led by Jill Banfield's lab. And Jill is one of the leaders in metagenomic DNA analysis, which means looking at DNA that comes from microbes that have never been identified by scientists before, have never been cultivated in the laboratory, and are analyzed simply by collecting samples, sequencing DNA, and then assembling genomes and looking at what kinds of interesting pathways and viruses and things like that occur within these samples. And so through doing work with Jill's lab, we've been investigating the occurrence of different kinds of CRISPR-Cas systems in microbes. And one of these that came to light that we first published on back in 2017, which in the CRISPR field is sort of a long, long time ago, is an enzyme that we called Cas-X. And so this is a protein that was identified by David Burstein, a computational scientist working in Jill's lab and mine. And the way he did his analysis was to go through metagenomic data looking for genes of unknown function that occurred in the neighborhood of a CRISPR array, which is the signature sequence of CRISPR adaptive immune systems. And importantly, this Cas-X protein and other proteins like it that we've identified for study are enzymes that don't have identified functions. So in this particular cartoon, the three genes colored in, shown by those blue arrows, are all genes well known to be involved in integration of new sequences into the CRISPR array. It's the adaptive part of the immune system. And then there was a fourth gene that we called Cas-X, which was of undetermined activity. And so what does one do with something like that? How do you figure out whether this gene is interesting and what it might be doing? And so we had a hypothesis that this would be some kind of RNA-guided protein based on the fact that it had one identifiable nuclease domain called ROV-C, which is a domain found in all of the other enzymes involved in CRISPR pathways so far that are involved in RNA-guided DNA cutting. And so I wanted to just briefly show you two kinds of assays that can be run to test for functionality. The first is using the enzyme, in this case Cas-X, in a format where it doesn't cleave DNA but can bind. And so we mutate the active site of Cas-X, which was identified by this, looking at the homology of this RUB-C domain to other similar proteins that have that type of nuclease domain. And then using the deactivated or D form of Cas-X in a bacterial test where we can analyze its ability to hold on to DNA at a particular place in the genome, and rather than cutting it, simply prevent expression of a particular protein. And so in this experiment, we have bacteria engineered to produce two kinds of colored proteins, green fluorescent protein and red fluorescent protein, and in the cells growing on that plate in the middle, on the left, when both proteins are made, the cells look yellow. And then when we turn on Cas-X in this format where it can bind but not cleave DNA and program it with guide RNA that can recognize the green fluorescent protein gene, what happens is that this protein has the ability to turn off green fluorescent protein expression and the cells look red. And then we can quantify that on the right compared to what we see for Cas9, and you can see that we see reasonably efficient silencing of the green fluorescent protein gene by Cas-X in this experiment. And then in mammalian cells, this is another sort of classic kind of experiment that people use to test for genome editing, and here we're using human embryonic kidney cells that are cultured. So these are cells that are growing in the lab in a dish. These cells have been engineered to produce the green fluorescent protein from a gene that's been integrated into the genome. And then we can program the Cas-X protein with guide RNAs that will recognize different positions in the green fluorescent protein gene to trigger an editing event that will disrupt the gene and therefore disrupt the ability of the cells to make the green fluorescent protein. And then on the right, we can quantify those data, and what you can see here is that on the top two plots on the right, we're looking at two different Cas-X variants that are naturally occurring with two different types of guide RNAs, and you can see slightly different efficiencies of these proteins depending on the variant that we use and the guide RNA that we're using. And then in the experiment on the bottom on the right, we're taking the cells that have been presumably edited and then growing them out over a number of days to show that those changes to the genome are permanent. They occur, they are maintained in the cells over time. So this shows that Cas-X is an active enzyme, and then the question is, how does it work? And so this is where biochemistry comes in. And so then what we do is we take the Cas-X enzyme and we can study its behavior in the laboratory by doing experiments like these shown here. So on the left-hand side, we're taking a guide RNA that's been, in this case, engineered into a single guide format, and using it to direct Cas9 to pieces of DNA that are quite short that we can radiolabel and test for cutting. And so in this experiment, we radiolabel the DNA, and you can see that over time, the DNA gets cleaved, and we detect that by applying it to a gel system where we allow the molecules to migrate through the gel. They move faster in this gel system if they've been cut, so they're smaller. So we can see efficient cutting going on. And then to figure out in more detail how that cutting is working, what we can do is to radiolabel each strand of the DNA and run a more sort of a higher resolution gel system to look at exactly how long the pieces of DNA are that result from this cutting reaction. In the experiment on the right-hand side, we do this, and in this case, we see that Cas-X actually makes a 10 base paired staggered cut in DNA. And that's quite distinct from what we see for Cas9, for example, which makes a blunt double-stranded break, and could be interesting for genome editing because it leads to a large overhang in the DNA that might allow different kinds of DNA repair to dominate in cells that have been treated with this enzyme, something that we're investigating at the moment. And then finally, because we're mechanistic types and we like to understand exactly how these molecules work, we've been working away on structures of Cas-X, and this is work done collaboratively with the lab of Eva Nogales, my colleague at Berkeley. And using electron microscopy, we've been able to solve a series of structures of Cas-X in different states of assembly with RNA and substrate DNA molecules. And here I'm just showing you one snapshot of Cas-X, it's the molecule on the right, compared to a similar structure determined for Cas9. And these are both structures where the protein has the guide RNA bound. The guide RNA is the bluish, kind of cyan-colored molecule, and then these enzymes are both bound to double-stranded DNA substrates, showing how they interact with the DNA duplex. And I just really want you to notice that Cas9 is interesting because it's quite a bit smaller in terms of the size of the protein compared to Cas9, but it's got a much larger RNA component to it. And so from a chemical perspective, these molecules of Cas9 have distinct properties compared to what we see for Cas9, and we think that's going to be interesting for thinking about how we deliver these molecules, especially as pre-assembled protein guide RNA complexes into cells for applications, including for therapeutics. So that brings me to the other little story I wanted to mention, which is thinking about once these molecules are introduced into cells for genome editing, it'd be great if we had a way to control their activity, not only in terms of how the DNA gets repaired, but also how long they retain activity. And this has led us to think about the way that nature does this, and to look into the presence of naturally occurring CRISPR inhibitors that are found, again, widely in bacteria. And so about 2 years ago, a wonderful postdoc came to my lab, Kyle Waters, who wanted to figure out how to look for these kinds of inhibitors systematically, which hadn't been done in the field at the time. People were sort of discovering these in a very stochastic way. And so what Kyle did was to ask whether we could identify bacteria that have to have inhibitors in their genomes because they've got CRISPR systems that would otherwise kill off the host cell. And so to do this, what he did was to investigate CRISPR systems that are found in databases now that have sequences that can target the host bacterial genome. And so we know that for those bacteria to survive, they have to have a way of turning off the CRISPR system, so they must have natural inhibitors present in the genome, and Kyle came up with a terrific way to identify those, and if you're interested, you can read the paper about this. And one of those inhibitors that he found was something that had been, first of all, was targeting an enzyme for which at the time there were no known inhibitors, and that's the enzyme called Cas12a. And when we started looking into how this Cas12a inhibitor operates, it turns out that it has a very interesting distinction from other natural CRISPR inhibitors, namely that it's what's called a multiple turnover inhibitor. So this is a protein that is able to interact with Cas12a and turn off its activity, and then it falls off and can bind to a new molecule of Cas12a and turn off its activity and fall off, et cetera. So it can do it again and again and again. And when we looked into how that was actually working, it turned out to have a fascinating mechanism, and I'm just showing you one snapshot here that illustrates how it works. So basically, this is an inhibitor that when you add it to the guide RNA molecule that's being used to direct the activity of Cas12a, you see no change in the RNA, and that's what you're seeing on this gel system here. And when you add Cas12a to the guide RNA without any inhibitor present, there's no change in mobility of the guide RNA. But if you add them both together, what happens is that the guide gets cut. And it's cut very precisely. It's not randomly degraded, but it's cut at a precise position. It's very efficient. And when we map those positions, what you can see on the right is a cartoon of the Cas12a structure showing that what this inhibitor does is it cuts off just the guide part of the RNA sequence. It leaves the rest of it there. So it's a wonderful way to turn off this enzyme by removing the guide. It no longer has the ability to target DNA, but it has the rest of the RNA molecule bound, so it can't easily rebind a new guide RNA molecule. Beautiful, right? It's just something that, natural selection has come up with. And so we think this is going to be a terrific way to turn off these kinds of enzymes. We've already shown that this is a very efficient inhibitor of Cas12a-mediated genome editing in human cells, and we're looking to employ this in the future as a way to control the activity of enzymes like this when employed for genome editing applications. So I'll just conclude by pointing out that new CRISPR enzymes continue to provide alternative tools for genome editing and genome control. And we think that by looking at natural examples of CRISPR inhibitors, like the one I showed to you, we'll find ways to control these enzymes in therapeutic settings as well. And I wanted to acknowledge a fantastic group of people I've had the pleasure of working with, a photograph here, their names on the bottom, as well as our funders. Thanks very much.
Now you heard wonderful sciences. So I want to invite the panelists come to the stage, and they will answer some of the questions. So I have 2 mics on each side, and see whether we have any. Questions.
Yeah, sorry. That's a bit of feedback. I'm Kathy Neocon. I'm at the Francis Crick Institute in London. I had a question for David Liu. So this is quite an ignorant question, but because the cytidine deaminases that you're using have an important role in DNA, in regulating DNA methylation, I wondered if you, or if you know of anyone else, who's evaluated any consequence of using base editing in terms of epigenetic changes at the target site, and whether that might have important consequences for how we think about the use of this technology, say in editing in human cells.
Yeah, it's a good question. The different cytidine deaminases that have been used for base editing now include variants that respond differently to methylated versus unmethylated Cs. So I suspect the answer to your question will ultimately prove to be that it depends on what variants you use. Some laboratories have recently reported the use of cytidine deaminase variants in base editors that accept much more willingly than our variants, methylated Cs, for example. And I'd say the substantial majority of cytosine deaminases that we've tried to use in a base editing context will function in that context. So the short answer to your question is that I don't think anybody has studied the epigenetic implications yet, at least in any kind of sort of more physiologically relevant context of base editing, and it's certainly an area that I would welcome greater investigation, but there's enough of a diversity of cytosine deaminases that have been used for base editing that I suspect the answer will depend strongly on the choice of editor.
So that may kind of answer the question in terms of efficiency of that base editor, but what I'm trying to ask is, as a consequence of changing a single nucleotide at a given site, does that have a consequence for example, imprinted genes and methylation at the target site, not just the specific base that's
Sure,
being
it's
changed?
certainly
Yeah.
possible. I mean, changing even a single base, regardless of the mechanism, can have that consequence on, can have that potential consequence on epigenetic regulation.
Thank you.
Hello, thank you for the wonderful talk. And I would like to ask 2 questions. First question is to all of the panelists. So the first question is, so Cas9 is now pretty much a well-developed platform for gene editing, but then there are still limitations to applying it in a clinical sense because of like inflammation, that, as mentioned by Professor Dobna. And although there are like investigation going on that to look into Cas9, Cas proteins that are non-inflammatory, but then how about like, is there any like potential delivery methods that are like that you highly suggest or like encourage people to look into? And then the second question is specifically to Professor Zhang, and this is about your recent work on the Cas13 protein. And could I get a brief overview on the comparisons between the different Cas proteins in relation to Cas13 and the limitations of Cas13 and the potential clinical applications and advantage of Cas13? Thank you.
I think we should start with Fang first. Answer the second question.
Okay. Thank you for the second question. So I didn't get a chance to mention about a different type of CRISPR enzyme called Cas13, so thank you for asking about it. So CRISPR are bacterial immune systems, and there are many different forms of CRISPR systems. In fact, nowadays there are 6 major types of CRISPR systems found. Cas9 is one of the 6, and then Cas13 is another very interesting system. The difference between Cas13 and Cas9 is that Cas13 targets RNA rather than DNA. And because of that, there are a number of different applications that can be developed using Cas13 to either knock down RNA inside of mammalian cells or to inactivate Cas13 so that it simply binds to RNA and then recruit different effector modules to specific mRNA or other non-coding RNA to alter their sequence function. So there are several different members of the Cas13 family. There's something called Cas13A all the way to Cas13D. And they are different in terms of their RNA, protein, complex structure, as well as the sizes and probably subtle differences in the targeting specificity of these different enzymes. In mammalian cells, where we try to translate the application of Cas13, it seems that Cas13B, one of the four subtypes, has the most robust activity. And one of the applications we explored is by using inactivated Cas13 to bring adenosine deaminases to specific RNA to be able to edit sequences at the RNA level rather than at the DNA level. There are various applications of Cas13. Cas13 can be delivered using either viral vectors or lipid nanoparticles into cells to be able to produce the editing machinery and also effect changes on RNA. And I think some of the most exciting applications might be places where you don't want to have permanent editing and you just want to have transient editing. There have been reports described that shows constitutive activation of genes in the Wnt pathway or beta-catenin can trigger regeneration and proliferation of certain cell types in the retina or the liver or hair cells from David's work. And engineering a gene like beta-catenin, you probably don't want it to be constitutively active because you probably get a tumor as a result of that. And there, by using RNA editing to transiently alter the phosphorylation state of that protein, you can have it be there for long enough to trigger the regeneration, but not long enough to cause a tumor. So that's just one of many applications that you could use Cas13 for. But thanks for the question.
Thank you.
And then anyone to take the first question?
Just go, please.
Oh, I was just going to comment on the first question. I think we all have advocated explicitly or indirectly in our talks the benefits of transient exposure to editing agents. So I think one of those benefits includes less of minimizing the chance that the immunogenicity, which is anticipated, given that, as Jennifer articulated, these enzymes come from bacteria that live on and in us, that perhaps transient administration, the one-time administration, might minimize the clinical relevance of that immunogenicity. There are, of course, many human therapeutics that are immunogenic. It's not mutually exclusive with being an effective therapeutic, but managing the consequences of that immunogenicity is an important consideration for the field. And I think getting in quickly, getting out, and never coming back is one approach that helps to minimize the chance that that immunogenicity reaches a clinical relevance that mitigates therapeutic benefit.
Thank you. And apparently, if you transiently express these proteins, you also reduce the risk of target activity, the cumulative risk of target activity during time. And regarding the different ways by which you can deliver these molecules, it really depends on the application. So if there are ex vivo applications in which physical methods are very effective and suited, in these specific cases, likely you don't even have immunogenicity of the proteins because again, they are transiently expressed, maybe ex vivo, and then implant back into patients. And transfections are very, very effective there, together with viral vectors. In vivo, apparently, for specific organs, some class of viral vectors are very powerful, adeno-associated viruses, for instance, but also, again, physical methods, lipid nanoparticles for specific organs are very, very suited for that.
Jennifer? All right, thank you.
Thank you for all your answers.
So thank you. My question is, could you please talk more about the unintended consequence using the genome editing technology? And is it possible to systematically evaluate or predict the unintended results when you're doing the experiment? And not only in or near the target region, but also for other locations in other chromosomes. And because of the news of yesterday, I think the layman like me, we really want to know what the side effect the twins will suffer in the future. Thank you.
So maybe Jennifer, the unintended consequences.
I had a very similar question as well. I mean, we spent an entire session on that at the 2015, and it's really not well represented here. So this is a very important opportunity to be talking about that, sort of understand the fidelity of these enzymes, and has our sense of comfort with the efficiency and fidelity changed in the last 3 years? And perhaps Angelo could comment on the fidelity relative to the known frequencies of insertional mutagenesis for viral transgenes.
Yeah, no, this
Thanks,
is a
Joyce.
very good point. So I'll start, and I'm sure my colleagues will have things to add. But I think, first of all, there has been a lot of work done over the last 3 years, so since 2015, on exactly this topic of fidelity of these enzymes. And I think from studying both the fundamental mechanisms of these natural proteins, but also work that's been done by, certainly by Feng and David's labs and our own and Keith Jung and several others, people have been investigating ways to engineer the enzymes to be more accurate than they are naturally. And so there's now quite a collection of these different kinds of proteins. There's even one that's sold commercially that I don't know if they even reveal what it is, but people in my lab use it and say, yeah, it's very, very accurate. So, I think that that's one thing to point out. It's just that the properties of these enzymes in terms of fidelity continue to improve. Secondly, I think it's important to point out that when we think about fidelity in a setting, sort of a clinical setting for genome editing, to me there's kind of a couple of ways you could define that. One is you get cutting at an unintended site, and that leads to an edit that we might call an off-target edit, something that you didn't want to introduce into the genome. And then there's also editing that occurs at a desired site, but leads to an unintended, undesired outcome, right? Whether it's from the type of repair that happens or just the fact that you've disrupted a gene that then has an effect that you didn't want or didn't appreciate. So I think those are both things that have to be addressed as the technology continues to advance and we understand more and more about how DNA repair is happening. And then, of course, I think, with both David and Angelo's work, these are applications of genome editing that are potentially very exciting and very interesting because they don't involve DNA cutting, right? So then you still might have the issue of off-target base editing or off-target epigenetic modification, but you're not triggering a covalent change to the DNA, or at least a change in the DNA sequence in the same way that you do when you introduce a cut. And what else? I think that it's very important to study the way that repair is happening in primary cells and tissues. A lot of the early work was done, and certainly when we were talking at 2015, in the 2015 meeting, a lot of that early work was done using cultured, transformed cells. And I think now a lot more work is being done in primary cells and tissues, organoids, and then even sequencing in whole in animals and looking at off-targets that happen in those settings. And I think more of that needs to be done. And then finally, related to a conversation I had earlier with Victor Zhao, I think it's going to be incredibly important for the field to come up with ways of standardizing the analysis of off-targets. Right? That still really hasn't been done. I have to give Keith Jung a lot of credit. I think he's been trying to kind of lead the charge on this, and I know that NIST and several people here have been involved in those conversations, but we need to accelerate that. I think that's really key, especially when we think about clinical applications. We need to have ways of comparing in different experiments off-targets, deciding, what do we consider an off-target, and, what level of off-target event is acceptable in a clinical setting. So.
Just one point regarding these affected domains that we are using to modify the epigenome or the genome. As said before, we are not most of the time delivering a break, so we are not using non-homologous unjoining, but these are effector proteins that have biological activities, and we are used to express them at high level, by transiently for sure, but at high level. So again, off-target activity also in this context should be thoroughly assessed. Isn't easy as in the case of genome editing, because at least in the case of epigenetic editing, you may impose new epigenetic mutations that might be more difficult to be found than genetic alterations. But it's something that anyway have to be analyzed. Coming back to the point of standard gene therapy versus gene editing, if you are considered that you are injecting millions of stem cells into patients, so probably millions of different random integrations, and that these vectors can often monoallelically modify genes, so they may cause haploinsufficiency or upregulation of only one allele. It is likely that by using gene editing, having very high efficiency of editing, we can achieve by, in terms of disruption, efficiency of 80–90% in terms of disruption, and possibly 1–2% of target sites. I think that we have to carefully assess that. So gene editing ex vivo, trying to understand the off-target profile of these molecules that are really reagent bound. So in the case of Cas9, probably guide bound. So it really depends on the guide that you are using. Apparently, people are now exploiting analysis, bioinformatic analysis, to predict guide RNA with very good specificity. So then they might be the beginning of, but then you have to do your experiments and assess of target activity of the endonucleases.
Maybe I'll just add that I think those are great summaries of a lot of issues surrounding undesired and off-target editing. But I think the consequence of off-target editing that is viewed as acceptable or unacceptable will depend strongly on the disease being treated. For me personally, I worry predominantly about one type of off-target edit, the type that causes uncontrolled cell proliferation. If in a small percentage of edits, one manages to disable a cell, let's say shut down translation, something that's essential for the cell, and that cell dies, that actually doesn't, I think, pose as much of a problem, doesn't make me lose sleep at night. But I'd certainly worry if an off-target edit initiated cancer. Fortunately, I think that's a fairly bounded problem. There will be a finite number of genes and regulatory sequences that one could, in principle, keep track of and help guide our investigation of whether off-target edits are likely to be clinically relevant or not. I don't think there will ever be an editing technology that will be absolutely perfect in its specificity. But if we recognize that there is a basal mutation rate that we all suffer or enjoy, depending on your perspective, and that there are bounded, targeted ways to look at the consequence of off-target edits, those are important constraints. I've had parents of children with genetic diseases tell me that they would gladly trade even cancer for their child's, Rett syndrome or Dravet syndrome or progeria, et cetera. So it's also important to keep in mind that depending on the disease being treated, achieving targeting perfection may or may not be actually in that patient's best interest if it comes with slowing or withholding the development of a therapeutic that would already have the opportunity to improve the quality of their life. So all of these are complicated considerations that have to be analyzed carefully.
It's an important question. Yeah, I'll add one other thing. So there are kind of two ways to think about using gene editing to treat disease. There's using gene editing to treat genetic diseases, where we know what is a disease-causing mutation, we're going to try to fix it. Then the second way is to use genetics to treat common diseases. So two of the prominent examples that folks have talked about are CCR5 and PCSK9. CCR5 reduces risk for HIV infection, But unintended consequence is that it can increase risk for West Nile virus or susceptibility to the flu. PCSK9 reduces cholesterol, but unintended consequence is that you can increase the risk for diabetes. And so even when we have perfect on-target edit, there can still be unintended biological consequences that come as a result of introducing these common genetic variants. And that's another thing to really sort of think hard about before implementing any therapeutic strategy based on that.
Thank you.
Hi, my name is Santiago. So in the panel total, I saw like a few different metaphors being used. So starting with fungs, and then like you said, that you switched the scissors to like a pencil. And so I'm wondering what the function of these metaphors are for both stimulating research and exploring different ways of doing research, and also what the implications are for communicating your research and for discussing the social and ethical issues.
Metaphors.
Well, the reason I use scissors and pencils as a metaphor in a broad, mixed audience is frankly because it's difficult to communicate the essence of some of these technologies if you stick to strict molecular descriptions. So there's no motivation other than trying to convey the essence of what they do in a way that is maximally understandable, recognizing, of course, that if you maximize the fraction of the audience that will be able to get the gist of what you're saying, you probably are sacrificing some details around the edges. Base editors don't actually look like pencils, and Cas9 doesn't actually look like a pair of scissors, but there are functions that each of those classes of genome editing technologies perform that I think make those analogies useful for people who aren't specialists in the field.
No, I agree with David.
Is that satisfying? Yeah. Okay, thank you. I have one more,
Yeah, coming back to the off-target issue, there's some discussion around ways to reduce off-targets, but I don't think there's another opportunity to talk about the methods that are used to detect off-targets. And so I guess I'm curious from your guys' perspective, all of you, what do you think the detection limit is for finding off-targets right now with the existing technologies, whether it's in the context of a single cell for germline editing or hundreds of millions of cells for some type of cell product or in vivo editing, and if you have thoughts about quantitatively where you think the detection limit is now, where it might need to be, where it might go in the future, stuff like that.
Yeah, it's a great point. I wish we could Skype in Keith John, since he's made major contributions to methods development for detecting off targets. Jinsu Kim has also made contributions. He's here and other labs. I think from the perspective of these methods development projects, they're currently in a phase where maximizing sensitivity remains the goal. Even though I think with methods like CircleSeq, the sensitivity has now gotten to a point where hopefully Keith John won't see me saying this and conclude that I've misrepresented his work, but that he recognizes and has said in his lectures that there will come a point, maybe it's CircleSeq or maybe it's another generation of technology, where the sensitivity is so high that you may not that everything that you pick up may not actually be relevant for research
but
or
still,
therapeutic editing.
David, so for like FDA to approve your product, they have to have access to QC, QA, whatever you have. So even though you are 100% sure, they say you have to do it to...
Yeah, that's right. And in my experience, the FDA regulators are actually very forward-thinking and realistic about the need to balance these considerations. They appreciate...
They would demand data, other than...
That's right. So you need real data from real methods that have been validated by researchers. And I think they've already commented on the presence or absence of such data in NDA applications.
So David, do you think it'll be possible to take a technology like CircleSeq, for example, or something similar, and come up with a standardized way to, basically determine off-target levels?
Yeah, so an Ames test for genome editing, as one likes to say.
Yeah.
I think that's an important goal. I wish there were more people working towards that goal, I agree that I think that would actually be an important step forward for the field. And I'm grateful to those like Gee's lab and Jinsu's lab and others who are putting the pieces in place to get there. I think there'll need to be a little bit of cultural change as well, in that currently during the methods development phase, laboratories tend to use methods that they develop because nobody else will showcase the strengths and weaknesses of their methods if they're very new methods. So increasingly, my lab, for example, is adopting methods that others have adopted and shown to be effective at identifying these off-targets. Even though, of course, we had nothing to do with the methods the development of those methods, but I think towards this goal of having a more standardized workflow, an intellectual workflow that we all accept, that can be used to evaluate off-targets in an apples-to-apples kind of comparison, that doing so is important.
And I think another important thing to keep in mind is, as we assess specificity of nucleases or base editing specificity, we have to move beyond hex cells. So much of the study is being done on 293 cells, and they have their particular organization of the genome, their particular epigenetic state. Primary cells are likely to be different. So the more we can push these technologies to make it possible to detect off-targets in primary cells or even primary tissue, I think the more we'll be able to learn about the specificity of these systems. And then the second thing is, at some point, we're going to, the sensitivity of an assay is going to be competing against the natural spontaneous mutation rate of the cells. Especially working with cells that are rapidly proliferating, there's going to be some rate at which they accumulate spontaneous mutations. And so being able to sort of accurately identify a mutation as an off-target editing-induced mutation versus spontaneous mutation is also very important. And I think, I think that's an area that still needs a lot of development.
If I may.
All right. So go ahead.
No, if I may agree with Feng. So I think GuideSeq is already quite enough sensitive. We might risk actually of finding more mutations that were actually then present in the cells of interest. And we have to test also the reagents in cells, relevant cells, but not only because of target activity, possible off-target activity of these enzymes, but also because these cells may react differently to the different procedures that we are doing. Activation of the DNA double-strand breaks response, etc., etc. These are again molecules that might have biological consequences on the cells that we are manipulating. So I think that we have to assess also such kind of, let's say, unwanted effect of the technology by maybe transplanting the cells into animal models and assess their functionality.
All right. So can you be very brief?
Okay. Well, my name is Samson Lau. I work for Delos Capital, a Hong Kong-based venture capital fund invested in life science. My question is for Professor Doudna. I recently came across a comment that because of the source of Cas9 enzyme, we will see more and more anti-Cas9 response in human going forward. So going forward, the new editors, Cas-X and Cas-Y, discovered by your lab and Professor Banfield's, will become more and more important. What would be your view or response to such a comment? Thank you.
So as I mentioned in my talk here, the enzymes that have been used primarily for genome editing experiments to date have come from pathogenic bacteria. So these are bacteria that probably many of us have been exposed to over our lifetimes, and many of us have pre-existing antibodies to the proteins that are found in those bacteria, including Cas9 and related enzymes. So that creates a potential problem for use of these proteins in a clinical setting. And so what I think is there's certainly multiple ways to think about dealing with that, but one possibility is that we can deploy enzymes that are coming from non-pathogenic bacteria. So Cas-X and Cas-Y are examples of that, but there actually are also quite a wide range of Cas9 and maybe even Cas12a variants that are coming from non-pathogenic bacteria. So potentially those will also be, can be employed to get around the problem. And then, as you may know, there's a lot of work going on in other fields, companies that have been developing biologics, which are based on antibodies that have been humanized, and, a lot of thought has gone into dealing with immunogenicity of molecules. So I think that that's also, some of those concepts could be applied for these gene editing molecules as well. So I think there's a lot that can be done. It just hasn't happened quite yet.
Thank you very much.
This one?
Hi, my name is Jonathan, and I'm a student in computer science, but study a little bit molecular biology before. So that's why the problem of search has been very interesting to me. And it seems like today's there has been a recurring theme among speakers, which is the motivation of looking into the natural diversity of microbiome family. So be it, for example, characterizing the full diversity of the caste system, or using evolution strategy to find more different types of enzymes. And so it's like this question direct to all speakers. It's like as a molecular biologist, what are some of the areas that you think is worth looking into in the context of genome editing? Like what type of other tools we should expand in the toolbox? And then the second question is, do you think the field has kind of converged to a way to fully search the whole possibility of this wonderful microbiome system? Family, yeah. So I would love to hear what you guys think about.
This is a fun one.
In a very brief sense. Yeah.
Brief.
Yeah, it doesn't have to be details.
Well, the nature is a big treasure trove. There are a lot of interesting and powerful systems to be found in nature. I think there are probably two main areas for genome editing therapy that need development. The first thing is, how do we introduce DNA into the genome in an efficient, precise way that doesn't depend on cell replication? Homologous recombination depends on active cycling of the cells. So if we want to fix and replace DNA sequences, we need something that can also work in non-dividing cells. The second area is delivery. How do we introduce these editing machineries into different tissues in the body, different cell types? How do we do it in a tissue-specific way? I think if we look into nature, there are probably solutions to some of those problems in the natural diversity, and you should look into that.
That's a good question. So in the interest of time, we still have two from the audience asking questions. Go ahead.
Ben Hurlbut, Arizona State University. I wanted to pick up on Dana Carroll's important comments earlier about equity and access. And Jennifer, you mentioned a proprietary protein, or proprietary enzyme. And I just simply wanted to ask whether and how, given the focus on application and therefore the focus on industrialization ultimately, how issues of proprietary interest are shaping both the scientific landscape and how considerations of access and scalability are or are not entering into research strategies for exploring the kinds of tools that you all discussed, and whether there are things that could be done differently or better.
Jennifer?
So Ben, thanks for that question. It's a really important point. I'm really glad you brought it up because it's something that I think about a lot, and it hasn't really come up too much. Maybe there was one or two talks this morning that brought up this, sort of point about equity and access to, this technology. And I think it's something that is very difficult to address. As Dana Carroll pointed out, I thought very, very nicely, saying that this is something really important to think about, but it's hard, right? It's hard. And so the moment I think all of us, well, I'll let my colleagues maybe speak for themselves if they're doing something different, but I think we're all of us that are sort of more on the technology development side of things are, racing forward with that. rather than really thinking about, how do we ensure that there's going to be widespread access to this later on. But it's, it is a challenge to tackle. With respect to proprietary enzymes, I think for those of us, certainly for those of us working in academic settings, there's no prohibition on using these proteins. We can freely get access to any of them and work with them in the laboratory, which is great. And, this is one of the things about the CRISPR-Cas9 technology that's frankly so wonderful, is that it's widely available. It's been accessible by people around the world. So that's really driving the research enterprise right now, because basically people can get access to this from organizations like Adgene that distribute these molecules very inexpensively, so that people can, the technology is not limiting what people can do in their labs. And, and I think going forward, I sort of imagine what will happen in the future is that you can already see more and more of these enzymes being developed, right? And eventually there'll be a very large, toolbox of proteins that have, somewhat different functionalities from each other. And I kind of imagine, it's like the old days when I was in graduate school. We used what was called the biolabs catalog, had hundreds of enzymes in it. Now it's going to be the sort of that equivalent for gene editing molecules, and I think that we'll have the ability to basically pull the tool off the shelf for the application that's needed. And again, in an academic setting, those will all be widely available. In commercial settings, that's something that's still being sorted out, is how will companies get access to the tools that they need for particular applications. And that'll take time. And I think it's playing out partly in just the fact that as more and more of these enzymes become available, it just, it does make it easier to get a hold of those that are going to be most effective for a particular application. But I don't know if there's one.
Let's take the last question.
I was a little bit surprised to hear that when you gene edit PCSK9, then you increase the risk of diabetes. There are two actually approved product anti-PCSK9 antibody on the market, one from Amgen and one from Regeneron. And as far as I know, there is no report saying that using those antibody to lower cholesterol lead to increased risk of diabetes. What do you think about that can account for the difference? My second question is that we talk a lot about off-target effect, which is certainly the major hurdle for CRISPR application. What about mosaicism? For example, in the progeria model you only have 27% of your cells getting back to normal lemon, right? Which means that probably 70% of the cells do not. So how does it affect the outcome? For example, are the mice suddenly the aging process get delayed or what? Yeah.
Okay. So fun. Diabetes.
Yeah. So I was quoting a study that looked at individuals who are naturally deficient in PCSK9. They live, healthy lives, but there is an elevated risk for diabetes. I think that's all to kind of highlight the fact that biology is complicated. There are a lot of intricate compensatory activities, so we should really, humility is a good thing. We should be humble and not change too much.
Good, thank you.
So with respect to your question about the progeria mouse model, while it's true that in all the organs we've looked at in vivo in this early case, we see editing that varies up to 60-some%, but it's never 100%.
But David, you only have one shot. If you repeat, the second round and the third round, would I increase the level of efficiency?
Yes. So I'd say we're in the process now of first trying to optimize the result. That was actually a pilot experiment where I was expecting maybe 1 or 2% editing, and we would start from there. So I think it's a promising start, but we still have a ways to go. But second, I think the extent of editing that you will be needed to offer a therapeutic benefit to a patient will depend a lot on the organ, of course on the disease. In the case of progeria, for certain organs that are more regenerative, like the liver, it's possible that even editing 60% of the liver will actually be enriched. And I think researchers have already observed that in other animal models where editing the liver, for example, to treat tyrosinemia, you can get a small percentage of editing that eventually becomes amplified as the non-edited cells are replaced by edited cells because they have a growth advantage. The cells that actually express progerin are sick compared to normal cells, so it's possible that an advantage like that may also exist in progeria, but a lot of research is needed to define what the consequences are, both in the animal models and prospectively in patients, of creating organs where a certain percentage is now fully wild type, another percentage is fully uncorrected. And that's very different than traditional therapeutics, where we take a pill and maybe we suppress 80% of activity in 100% of our cells. So this is really a different kind of correction that we now have the opportunity to begin to characterize.
Right.
But
Well,
that
progeria is
characterization
a
is
complex
important.
case. What about the simpler case that Angelo mentioned about the PCSK9? You try to silence PCSK9, right? I assume it's not incomplete also. Does it lower cholesterol? Does it lower LDL?
Well, these were experiments in cell lines in vitro.
In vitro.
We haven't measured cholesterol, we measured the expression of PCSK9 that was brought to zero. If you want to go to in vivo, it's more complicated because you have to express 3 proteins, so 3 repressors plus guide RNA. So we are developing the non-viral delivery system to do that in vivo.
Okay, thank you.
So with that, I'd like to thank the speakers, and I thank the audience too. Thank you.